Okay, great. Well, thank you very much, Christina, for the opportunity to take part in this series of Butterfly Talks. And thank you to all of you who are joining today. I'd like today to share some reflections on butterflies, microclimate, and climate change. I'm going to start off talking about how the distributions of species or their geographic ranges are linked to climate and what the responses of those distributions are therefore expected to be to climate change. But then I want to demonstrate that in reality the picture may be rather more complex because of geographic variation, both in microclimatic conditions and in rates of climatic change. And because of this microclimatic variation, I want to present some of the evidence of how it affects the processes that are involved in range shifts, namely colonization and local extinction, which are the key processes that are driving range expansions and range contractions, respectively. And finally, I want to conclude by just considering very broadly how some of this understanding might be useful or important for adapting conservation to climate change. Now, I suppose starting at the beginning, one of the first experiences that many of us have as naturalists is opening a field guide like this one by Higgins and Riley on the butterflies of Europe, originally published in 1970, and finding a representation of the distribution or the geographic range of a species. So here's two examples. Here's Melitea sphinxia, the glanville fritillary on the left, that we can see has a latitudinal distribution from the central Iberian Peninsula and the Mediterranean basin up to the English Channel and the Baltic Sea, with a few areas of distribution out here, for example, in the Atlas Mountains as well. So we have clear sort of latitudinal limits to the distribution of Melitea sphinxia. And then maybe we have a distribution map such as the one here for the Apollo butterfly, Parnassus Apollo, showing this much more patchy distribution associated with areas of high elevation in the mountain ranges of southern and central Europe, and also perhaps with in some lowland regions towards the northern limits of its distribution in Scandinavia. Now, of course, when this field guide was published, it was before the most recent period of climate warming really began. But of course, by the turn of the 21st century, we had strong evidence for anthropogenic climate change. And so a lot of ecologists and biogeographers were interested in quantifying the associations of species with climatic conditions, and therefore how the geographic ranges of species might respond to climate change. So these two maps shown here, again for the same two species, are taken from Joseph Settele et al.'s Climatic Risk Atlas of European Butterflies that was published in 2008. And the black dots on these maps show the 50-kilometer grid squares where each of those two species had been recorded. And the orange shaded areas show the climatic conditions that correspond to comparable conditions to those in the places with records of those species. And we can quantify the climatic associations of species in a number of ways. One way is by taking the average annual temperature in each of those 50-kilometer squares where the species occurs, and summarizing that as the species temperature index, which you can see here is rather higher, 9.6 degrees C for Melitea sphinxia than it is for Parnassus Apollo, being restricted much more to mountain regions. So one of the species I'm going to speak about a lot today is the silver-spotted skipper butterfly, Hesperia comma. And you can see here that the species temperature index for Hesperia, is intermediate between Melitea cinxia and Parnasius Apollo. So it has a patchier distribution than Melitea cinxia down here towards the southern limits of its range. But it occurs much more in lowland regions than Parnasius Apollo does, for example. And the two parts of the world that I'm mainly going to be speaking about today are this area here in southeast England, which I understand today has pretty sunny conditions, and where I'm joining you from today, down here in central Spain, which for the last few days has been pretty cloudy and wet. Now, of course, butterflies are ectotherms, so their body temperature depends on the temperature of their surroundings, and therefore their activity as adults is influenced by the temperature conditions when they're active, but also their growth and their development through their juvenile stages is influenced by the temperature of their surroundings. And of course, weather conditions also influence the condition of their larval food plants. And therefore, that we expect that when the climate changes, when we get changes to temperature and precipitation, we expect changes to the population dynamics of species, and we expect changes to their geographic distributions, potentially with colonizations towards the sort of cooler limits of their geographic ranges, and potentially with local extinctions or range contractions near to the lower latitude or lower elevation limits to their geographic ranges. And in fact, butterflies were one of the first groups that provided strong evidence that species were shifting their geographic ranges in the ways you would expect under a warming climate. So here's one example led by Camille Parmesan et al. From 1999, based on the distributions of European butterflies, showing for a number of non-migratory species that there were range expansions at the northern limits of their ranges. So here, the silver-washed fritillary Argenis pathia colonizing into Scandinavia, and the sooty copper Lycaena tityrus colonizing here into Estonia, and some evidence of range contractions towards the low latitude limits of their ranges, such as here in Algeria for Argenis pathia, and here in Catalonia for Lycaena tityrus. We also expect changes to the elevation ranges of species. And so some work that we did here in the Sierra de Guadarrama mountains in central Spain, we were lucky enough to be able to look at some data on the elevational distributions of butterflies that was collected by a scientist, Victor Montserrat, in the late 60s and early 1970s, and then to revisit those mountains in 2004 and 2005. And over that period of time, the mean annual temperature had increased by 1.3 degrees C. And we found that for 16 mountain butterfly species, on average, the lower elevation limits of those distributions had shifted uphill by just over 200 meters, which is a very similar rate of uphill shift to the rate at which isotherms were shifting up the slopes of those mountains. And we also found some evidence that the species were colonizing at higher elevations in the mountains as well. So we get this kind of pattern that's been referred to as an escalator effect, where species that are perhaps associated with warmer conditions, like or at lower elevations like Melitea cinxia, are shifting their distributions up the slopes of the mountains and replacing some of the species which might have previously been associated with those conditions, like a cooler associated species like Parnasius Apollo, which is being more and more sort of pushed towards the tops of the mountains. Now, we now have many examples of these kind of poleward and uphill range shifts across many different taxonomic groups and associated changes to the composition of ecological communities. But of course, as with many things or most things in ecology, it's not quite as simple as that, because climatic conditions are not uniformly distributed over latitudinal and elevation gradients. So obviously, if the Earth's surface was completely uniform, we might expect that at a higher elevation or a higher latitude, we have the highest precipitation and the lowest temperatures, and at a low latitude and a low elevation, we have the highest temperatures and the lowest precipitations. But of course, because the Earth's surface varies in terms of its topography, in terms of the vegetation covering a particular location, actually moving from the low latitude limit of a species range, such as these mountains here for Hesperia in central Spain, up to the higher latitude limits of the species range, such as this chalk grassland here, more or less at sea level in southern England. There's lots of variation in slope, aspect, vegetation, the rockiness of the terrain. And so for species which are occurring in both of these kind of locations, like Hesperia and Aglaia urticae and Pieris napi, the temperatures that they are experiencing at the different stages of their life cycle may not be easy to represent in terms of a latitudinal gradient of how favorable those conditions are for those particular species. So clearly the temperatures that are experienced by a butterfly or by a larva feeding on a herbaceous plant are going to be influenced by the elevation of a location, by its slope and its aspect, but also at a much finer resolution by the vegetation cover and by microtopography. So in these two thermal images at the bottom of the slide here, we can see that on an area of heathland on the Lizard Peninsula in southwest England in May 2013, around early afternoon, that an area of bare ground on the surface of the ground can be something like 20 degrees C warmer than a shaded area in amongst longer vegetation. And clearly all of these kind of different scales of variation in temperature conditions are going to influence these ectothermic organisms like butterflies. We also get variation in the rates at which the climate is changing. So in the left-hand map here, you can see the rates at which summer temperatures have been warming in England and Wales. We can see that in lowland, southeast England, such as this area of chalk grassland here, which is just on the southern edges of London, the temperatures have warmed over the course of 30 years by 1.5 or more degrees C. But the rates of summer warming tend to be less than 1 degree C if we go to more upland areas or coastal areas in particularly on the western fringes of England and Wales, such as this area of bracken and woodland here in Dartmoor, which is down here in southwest England. We go back down to the Lizard Peninsula, which is shown in this black box here. We can also see much finer resolution changes in the rates at which the climate is warming in ways that are relevant for insects. So here we see in the top two panels that the rate that the summer temperatures and the length of the growing season have been increasing much more rapidly towards the western coast of the Lizard Peninsula than on the eastern coast. But in contrast, if we look at the frost-free season, so the time for which young stages or food plants are not exposed to frost, the frost-free season's actually been increasing more on the eastern side of the Lizard Peninsula than it has on the western side of the Lizard Peninsula. So there are different effects potentially happening during the summer versus during the winter. And this is resulting from prevailing weather conditions and the fact that it's more cloudy on the eastern side here, and there are more sort of sunnier conditions on the western side here of the Lizard Peninsula. So this kind of finer resolution variation in climatic conditions is very important. We also see that the rate at which conditions have warmed over time might actually be less than the spatial variation there is in microclimate over a much smaller scale. So again, looking at the rates of warming up to about 2 degrees C, but just moving around on the Lizard Peninsula, there's something like a 5 or 6 degrees C difference on a summer day between the coolest locations and the warmest locations. So if there's suitable habitat available, this might allow species to redistribute themselves on a much more local scale in order potentially to be able to cope with the kind of rates of warming that are occurring. So what I want to do now is to actually look at some of the evidence that this kind of variation in microclimate could be important in understanding how species distributions have changed over the course of recent climate warming. And I want to do this by looking at the key processes that are involved in range shifts. So particularly at high latitude or cool range limits, looking at how microclimate could influence rates of colonization through its effects on the activity time that's available for butterflies, and also on the growth and development rates of their young stages, and also in terms of just how ambient temperatures might influence the availability of suitable microhabitats in the areas of habitat in and around where populations of a species occur. And in contrast, I'm going to look at how microclimatic variation could influence the rates at which species are suffering local extinctions where conditions are becoming too hot or dry for them, by varying how exposed they might be to unfavorable conditions or influencing the distances that they might need to move to maintain themselves in suitable conditions. So first of all, I'm going to talk about Hesperia comma at its northern range margin in southern England. So this is a species which, like many other butterflies in England, is at the northern limits of its range and therefore is associated with what we would consider hot microhabitats. So Hesperia comma occurs predominantly on south-facing areas of chalk grassland with short vegetation and a lot of bare ground. And these provide warm enough conditions for it to complete its life cycle. This species suffered a really serious decline in the mid part of the 20th century, partly because of the abandonment of extensive livestock grazing, but also because of the introduction of myxomatosis to the rabbit population in England around the mid part of the 20th century, which resulted in the vegetation getting much longer on many of these sites, and many of these sites being completely covered in scrub. So the species retracted back to something like 50 or 60 populations across 5 or 6 different ranges of chalk hills. And so when I first was going up onto those chalk hills in the 1970s, it would have been very, very difficult for me to find Hesperia comma in those sites. Some other things that started changing around the 1970s were that the rabbit population started to recover, and the conservation organizations started introducing conservation grazing on a number of these sites and undertaking management of the scrub, removal of the scrub to recreate some of these short turf grassland habitats for many of the invertebrates and wildflowers that had suffered because of these changes in vegetation structure on these sites. But also what happened around the 1970s was that temperatures started to increase. Now, we're lucky in that Chris Thomas is shown here along with his brother Jeremy and David Simcox undertook a complete survey of habitat for Hesperia comma and populations of the species across southern England in 1982, which means that we've got this evidence of where populations of the species were back then. They're shown in dark blue here, and where habitat was, which you might be able to just about see as these kind of pale sort of green symbols here. And this survey of the distribution of Hesperia comma and its habitat has been repeated every 9 years since 1982. And Crispin Holloway, who's with us today, he's been involved in 3 of those surveys. We did another one in 2018. John Benny has run the last 2, and Jenny Hodgson and Callum Lawson were also involved in some of the research I'm going to talk about here. Now, microclimate is really important for understanding how Hesperia comma has been able to expand its range through many of these ranges of chalk hills over that period of time. So you can see that these dark blue dots showing where the populations are have been sort of expanding out from where their sort of refugial populations were back in 1982. And this expansion has been particularly marked down here in Sussex in a range of hills called the South Downs. Okay, so why is microclimate important? Well, first of all, this butterfly lays its eggs really in the warmest microhabitat. So here are some eggs. They're laid on plants like this Festuca ovina tuft of grass growing in amongst areas of bare ground on south-facing slopes. In 1982, this butterfly was really restricted to laying eggs on areas of south-facing habitat with lots of bare ground. We see that when we look on different sites, on the hottest sites, the steepest south-facing sites, they don't have such a strong requirement for laying their eggs next to bare ground. And we also find that since 1982, the association of those eggs with bare ground has also reduced. So here's some data from a site in a range of hills near London called the North Downs, where in 1982, Chris Thomas did a bunch of quadrats for the eggs on 2 chalky banks on those sites. And Chris found, these are the red bars, that most of those eggs were found where there was something like a 30 to 50% cover of bare ground. But when we repeated those quadrats on exactly the same location in 2001, we found that the highest densities of eggs were actually in areas with around sort of 5 to 10% cover of bare ground. And this kind of association with a lower amount of bare ground has continued when we've redone that survey 2002, 2009, and later years. As part of this study, which was led by Zoe Davis during her PhD, we also found that the rate at which Hesperia comma lays eggs is influenced by the temperature at ground level on these sites. So we need an ambient temperature of something like 25 degrees C for those female butterflies to be active and to be laying eggs. Why is this important? Well, if we look at a south-facing slope and a north-facing slope on a sunny day in this area, then we tend to find that the south-facing slope is something like 10 degrees C warmer than the north-facing slope, which might be really critical in terms of creating these threshold temperature conditions for Hesperia comma to be active. Now, we know that summer conditions have generally warmed since the 1970s and 1980s, and this, of course, influences the condition within these sites. So if we look at a year like 1983, and this is a model that John Benny did for the number of hours that would have exceeded that 25 degrees C threshold across a number of across different sites through the distribution of Hesperia comma. This site, this is a big south-facing site called Deep Dean, which is the one population in the South Downs in Sussex which actually survived all the way through the 1970s and the beginning of the 1980s. And therefore, the population that survived on this site, which was the hottest site, which had the largest number of hours above 25 degrees C in 1983, it's individuals that have expanded out from that site that have recolonized the rest of the areas of suitable habitat for Hesperia comma in that region. You can see that many of these other west or north-facing sites in that region in 1983 had very few hours when it would have been warm enough for the activity of that butterfly. If we look at the region through which that species, Hesperia comma, has expanded its range, and this is kind of the fringes of the distribution in 1991. By 1991, it had, so Deep Dean is further away to the east here, where I'm pointing the arrow there. By 1991, it had colonized these two areas of habitat here. This line here shows the south coast of England. So one of the first sites it colonized was this steep south-facing nature reserve here, which in the 1980s tended to have a large number of hours above that 25 degrees C threshold. But if we look at the sites that it then colonized over the course of the following 15 to 20 years, during the 1980s, most of these are north-facing sites. They would have been much too cool for this species. But between 2000 and 2006, the average number of hours above 25 degrees C on those sites was much greater because summer temperatures were warmer, and the species was able to colonize all of these sites by the time that we did those latest surveys in 2000 and 2006. So the hotter summers increase the carrying capacity and the activity time in these different areas of habitat, and they therefore create more potential colonizers, and they allow the species to spread more quickly through the landscape. And it's through this kind of link between ambient conditions, the microclimate in the different populations, and therefore the rates at which the species can colonize, that we can see how fast the species is expanding its range. So in these three graphs here, the bottom graph is showing the August temperature anomaly in the different years between 1982 and 2006. This middle graph is the change in the National Population Index for Hesperia comma. So this is on regularly monitored sites from the UK Butterfly Monitoring Scheme. You can see that in the hottest years, like here, 1990, we have the highest population density on sites. And then the top graph here shows the results of a meta-population model that we did, where we modeled the rate at which the species was colonizing through the system based on the different microclimatic conditions in those sites. And we can see that the rate of colonization of new patches is highest, we expect it to be highest in the years when we have higher population on the monitored sites and we have the highest temperatures. So this is kind of the link between ambient temperatures, microclimate, and the rate at which a species is able to extend its range under a condition of warming temperatures. So that's how microclimate can influence range expansion, but obviously from a conservation perspective, of course, it's also very important to consider how microclimatic variation might help species cope with increasingly hot and dry conditions at their low latitude or low elevation range limits. And such as at locations here in the Sierra de Guadarrama in central Spain. And one great cause for concern in a changing climate is the Apollo butterfly, Parnassus apollo, which has gone from many of its lower elevation sites in recent decades, particularly in the Iberian Peninsula. So looking at that species in central Spain, in the Sierra de Guadarrama, well, one popular Spanish refrain about the weather in Madrid is that there are 9 meses de invierno y 3 meses de infierno. So of course, through its life cycle, the Apollo butterfly and also all of the other species that are occurring here in the Sierra de Guadarrama have got to cope with those 9 months of rather cold wintry conditions and those 3 months of hell that we get every summer. And you can see some of the wintry conditions here with Sarah Ashton conducting work for her MSc project on the distribution of Apollo caterpillars here in April 2007. Some of the results that Sarah found were that the temperature at the ground level where those Apollo butterflies are feeding was something like 3 degrees C warmer on bare ground, like in this photograph here, than it is beneath the shrubs that are also dotted over the sites where the Apollo occurs. Sarah also found that the temperatures were a lot less variable below those shrubs. And what we found with the Apollo caterpillars was that when the conditions were very cold, so less than 10 degrees C, and when they were very hot, above 25 degrees C, then those Apollo larvae tended to be underneath the shrubs in those sort of less extreme conditions. But at sort of intermediate temperatures, when the larvae needed to sort of raise their temperatures to be able to move around and feed on their food plant, which is this one here, Sedum amplexicaulet, then we tended to find them on the bare ground where the conditions were warmer. So clearly, having habitat which is available, which has got this sort of mosaic of both bare ground and low-growing shrubs, is very important for this species. And also we found that the Apollo tends to use more open microhabitats at higher elevations on the sites where it occurs in the Sierra de Guadarrama. So actually, you may need greater cover of shrubs down at lower elevations where conditions tend to be somewhat warmer. So this kind of understanding of how this fine resolution variation in vegetation could influence microclimatic conditions could be important in terms of managing habitat for a species such as this. Now, the temperatures in the Sierra de Guadarrama have gone up by in the region of 2 degrees C over the course of the last 5 decades. So between the 1970s and 2004 and 5, when we revisited a number of sites over the elevation gradient, the temperatures had increased by 1.3, 1.4 degrees C. And then since then, we've got a continuing increase in temperature over this mountain range. So clearly, the populations occurring here are being exposed to quite rapidly changing thermal conditions. And we found that when we revisited a number of those sites in 2017, that the majority of species were continuing to generally shift the average altitude at which they were occurring further up the slopes of those mountains. And generally, we had stronger evidence of local extinctions towards the low elevation limits of those species ranges than of colonizations towards the upper limits of those species distributions. So what sort of mechanisms might be driving these low elevation extinctions and these range shifts overall? Well, one of the species that we've looked at that in is the black-veined white, Aporia crategi. And Richard Merrill here carried out some research looking at the location of egg batches and doing some translocation of egg batches for Aporia crategi on its food plant, the hawthorn, Crataegus monogyna. And one thing that Richard found was that the survival of those egg batches through hatching to young larvae and then through to forming a winter hibernaculum, the survival was greater at the higher elevation sites, and there was a very low rate of larval survival among egg batches at lower elevations, which suggests that something such as greater dryness or greater temperatures at those low elevation sites may be increasing mortality at the hotter or the drier sites for this species. One of the ways that the butterfly may at least partly aim to cope with this is by laying its eggs on the more shaded side of the host plants at lower elevation. So with the black vein white, the eggs tend to be associated with the south-facing side of the hawthorn and blackthorn shrubs, the higher up the slopes of the mountain that you go. So I mentioned early on about this idea of the escalator effect of species shifting their distributions up the slopes of mountains. And I mentioned this measure, the STI, the species temperature index, that you can base on the geographic distribution of a species to determine its kind of thermal association. Well, do we therefore expect that as species are shifting their distributions, that the average thermal association of species in a given community or a given location has actually changed, is actually increasing? So we can take the average species temperature index of species that occur at a site and calculate something called the community temperature index, or the CTI. So if we're finding that species that tend to be associated with low elevation sites and relatively warmer conditions, like these ones here at about 1,000 meters, do we expect that they are therefore shifting uphill and replacing some of the species associated with cooler conditions at higher elevations? And we can quantify this by looking at changes in this measure, the community temperature index, or CTI. Well, we've looked at this across the Iberian Peninsula at the scale of a number of 10 kilometer squares, and these black symbols here show the 10 kilometer squares that have been well sampled both before and after 1980. And we find that the temperature, the mean annual temperatures, increased in all of these 115 10 kilometer squares, but the CTI on average across these sites has not shown a significant increase. So we don't have evidence across the Iberian Peninsula that generally we're seeing at this kind of 10 kilometer scale and a change, this kind of systematic change in communities. But one of the reasons that we might be able to explain this is actually because of these kind of microclimatic effects, because what we see is that in the more topographically variable of those 10 kilometer squares, we get reduced rates of change, both in the climatic conditions, so we get less warming, and we also get a reduced rate at which butterfly CTI is increasing. So this could be because that topographic variability is reducing rates of warming, therefore reducing the exposure of species to warmer conditions, but it also could be because they are able to move around within those more topographically variable squares and therefore maintain an association with suitably cool or moist conditions. We also find these kind of effects of topographic variability on protecting species against climate warming in England. And this is, so just at the bottom of the slide here, I'm referring to some work that was led by Andrew Suggett, where we found that in more topoclimatically variable 10 kilometer squares, we get reduced risk of local extinction for a number of insect and plant species that are threatened by warming. So topographic variability could be really important in buffering species against the effects of climate change. So to conclude, the implications for conservation, well, maybe if we're thinking about where the potential refugial locations might be for species from climate change, we might need to think about the climatic variability within different parts of the landscape. So the places that are more variable, such as that have greater variability in July temperatures, or that have greater variability in solar index, which is a measure of how much the landscape is likely to warm up because of slope and aspect. These locations, these heterogeneous locations, by providing microclimatic heterogeneity, they might protect species against the potential negative effects of warming. We need to think about how our protected areas might fit into this kind of context. Large, high quality and potentially heterogeneous protected areas might be really important, both in terms of protecting species that are threatened by warming against unfavorable conditions, but also, as we saw with Hesperia, in allowing species to survive some of the unfavorable years that we get in a series of years that are obviously variable over time. But we also might need a number of small or more marginal areas of protected areas. They could also be useful in helping species to spread across the landscapes as the climate warms. So we might need these kind of stepping stones of small protected areas to help species spread their ranges. And in fact, Chris Van Swy was talking about exactly this kind of point when he talked about the recent research led by Johan Fourcade at the end of his talk 2 weeks ago. And finally, a kind of a more general conclusion at a continental scale. What I hope this talk really demonstrates is that we need to protect the variability that we have across the ranges of different species in terms of the habitats they occur, the different elevations and the different latitudes they occur. Because it's through protecting this variability that we're going to be able to adapt to change. And we need, therefore, to monitor populations across the geographic ranges of species and have conservation measures that are in place that allow us to understand how species are responding to climate change across the different parts of their distributions, and to respond as quickly as we can to the potential negative effects of climate change for conservation. So thank you very much. I think I've probably talked for way too long, which doesn't give me a chance to name all of the people who are listed on this acknowledgement slide, or to think of the people that I might have forgotten in the names that I've presented here. So thank you very much for persevering through this talk.